Good.
So it appears that we are officially live. Hello, everybody, and welcome back to another session of our Sussex Vision seminar series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's lab, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from Washington University in St. Louis, Professor Takeshi Yoshimatsu. Following his undergrad in bioengineering and chemistry from University of Tokyo, Takeshi joined Yukiko Goto for his PhD in neuroscience, identifying signaling pathways that regulate the maintenance of neural stem cell self-renewal. In 2008, he moved to the University of Washington in Seattle and the lab of Rachel Wong, where he worked on the development of retinal circuits in Zebrafish. In 2016, and with a focus on retinal color circuits, he joined University of Sussex as a postdoctoral fellow with Tom Baden, and late last year, Takeshi moved to US as an assistant professor and started his lab there at the University of Washington in St. Louis this time. At the same time, he's a visiting research fellow here in Sussex and a visiting associate professor in the BioRTC lab in Yobe State, Nigeria. In his lab, they are interested in cellular and molecular mechanisms during development, dysfunction, and regeneration. By focusing on the fovea and the surrounding area, the macula, and employing a wide range of tools and techniques ranging from molecular and imaging to behavioral, they seek to understand the processes that make a fovea developmentally and the ones that lead to degeneration later on in life. Personally, I'm very excited to have Takeshi here with us for his talk entitled Human and Zebrafish Retinal Circuits, Similarities in Day and Night. So without any further ado, from my side, please all welcome Professor Yoshimatsu. Takeshi, the stage is officially all yours.
Thank you very much for your very detailed and kind introduction, Josh. Good to see you. Okay, let me share the screen now. Hope you can see me. Actually, the other way around, sorry. Yeah, I was clicked the wrong one. I believe this is the correct one.
Yep, we're good to go.
Yeah, great. Yes, so yeah, I'm actually at the Washington University in St. Louis. It's very confusing, you know. I did a postdoc in University of Washington, and now I'm in the Washington University in St. Louis. Yeah, so I started my own lab last November, but then today I'm gonna the project I'm gonna talk about is all done when I was a postdoc in Tom Baden's lab, or actually even some of the talks is about the work I did in Rachel Wong lab in University of Washington. So I'm interested in, like as George mentioned, development and the function of the vertebrate visual system. And then I'm mainly using Zebrafish as a model system for to understand the molecular mechanism and how the development mechanism actually leads to the proper functioning of the visual system. So the motivation to use Zebrafish is nicely illustrated in this next slide, which somehow I cannot move. Okay, showing the evolutionary tree of vertebrate species and the photoreceptor composition in these species. So if you look at, let me actually use the laser pointer. So if you look at the mammals, for example, so I showed the photoreceptor composition of primates as example. As you know, like humans have three types of cone photoreceptors called the S cones, M cones, and L cones. And also there is one rod photoreceptor, which is for night vision. However, if you look at other vertebrate species, there are many species that actually have more than three cone types. And then probably it's kind of easier to notice that the major sort of like type is the major composition of the photoreceptors having 4 types of cones plus rods. So this is kind of represented by the fish, only fish, like they have like UV, blue, green, and red cones, as well as rod. And then same for amphibians, although they have 2 types of rods. Some species of amphibians have 2 types of rods. So there are some variation, like for example, reptiles, they have 4 types of cone, as well as other cones called the double cones. But there are 4 types of opsins. So then we thought that based on this photoreceptor composition, we can kind of like guess that the ancient vertebrates probably already have 4 types of cones, UV, blue, green, red, as well as rod. So then by looking at species that has the same photoreceptor composition, we thought we could understand how the retinal circuit are intended to, how the retinal circuit is designed originally. Then from there, we can understand how the, others, different, how the retinal circuit are diversified in different species. So then, importantly, not just the photoreceptor, although the photoreceptor composition kind of varies among species, the basic architecture of the retinal circuits are conserved across vertebrates. So this is kind of the shown as a diagram of the retinas on the side view. And then you can see that, always photoreceptors are in the outer retinal layer. And then there's 5 major classes of neurons, photoreceptors, horizontal cells, bipolar cells, amacrine cells, and ganglion cells. And then roughly the location of those 5 neurons are conserved across species. So then we can kind of, apply the knowledge we gain from the one species to other species, and especially in this case, zebrafish. Okay, so then I'm going to talk about 3 sort of like essential feature of the human vision and how these features are actually conserved in zebrafish. And then this is kind of the surprising thing we found as we studied the function of zebrafish vision, because we thought like, we thought we were gonna find something, different in zebrafish that is kind of represent the origin, like, or more ancestral design of the retinal circuit. But as we study more and more, we start to find that the feature of the vision in zebrafish is pretty much many features of the vision in zebrafish are shared with humans. So that means actually the basic structure or function or designs are still conserved. So the 3 features I'm going to talk about is the color vision. So this is kind of the figures I took from the old papers about the human vision. So like in the human, we have 3 types of cone photoreceptor. Each has a different sensitivity peak to a different wavelengths of light. And also the sensitivity is different. And then there are some studies try to understand how these photoreceptors are used to sort of decode the spectral information. So it's about color vision. And then the best way to decode the spectral information using these 3 types of photoreceptors to have these 3 components. So basically one is so this is the y-axis is a response polarity. So 0 is indicated by the dashed line here. So then the best way to decode a signal from these 3 types of cone photoreceptor is to combine and then in a different way. But then there are 3 major components that would be the most efficient to decode the color information. And then these 3 components are illustrated by this solid line, which is the most primary one. And then the final dashed line, that's the secondary one. And then the third one is this more like a sparse dashed line. And then this is kind of the fundamental of how you decode the color. So the solid line is actually not about the color because always respond positively regardless of the wavelengths. Whereas the dash one, secondary one, they change the polarity of response depending on the wavelength. So this is actually encoding color, and then same for the third one. So this is kind of how we know about how human vision works. And then different aspect of the human vision is about high acuity vision, which is quite essential for our sort of vision we use in the daily life. As you may know, we have this special vision called the fovea, which is specialized for high acuity vision. And then from side view, we know like the structure of the retina is very different from the rest of the eye. So, and then the last three, this is, we also, those first two visions we use mostly in daylight, whereas there's another vision which is mostly used in night vision. And then this is quite extensively studied across mammals. So we know quite a bit about the circuitry. I'm not going to talk about this now, but I'm going to go into details later. So then for this talk, I'm going to talk about these two first very quickly because those are the already published work. And then second half, I'm going to talk about the night vision circuit in zebrafish and then how it's similar or different from human vision. So that's going to be the second part, which is still unpublished. Okay, so, and then first I would like to acknowledge people who are involved in the first part of the talk. So that was done in Tom Borden's lab. And this is a picture when I was in his lab. And then those are the people sort of involved. And then we also have collaborators for Philip Barron and Cornelius Schrader from University of Tübingen. And then also we collaborate with Rachel Wong in University of Washington. Okay, so the first thing we did to understand how the color vision works in zebrafish is to look at what is the role of outer synaptic layer of the retina, because we actually didn't know much about the color vision circuit in zebrafish, so we started from the first layer of synaptic layer. So the first layer of synaptic layer, we know what kind of neurons exist. So we know that there are 4 types of photoreceptors, cone photoreceptor in this case. And yes, I should mention that the words we particularly look at the larva zebrafish around 5 to 7, sorry, like 6 to 8 days post-fertilization. And at this stage of zebrafish, they don't still have words developed yet. So we only looking at the cone photoreceptors. So there are 4 types of cone photoreceptors, as I mentioned before. And then we know that there are 3 types of horizontal cells that are connecting cone photoreceptors in the type-specific manner. So, for example, H1 horizontal cell mostly connect red, green, and blue photoreceptors, whereas H2 connect to UV, blue, green, and then so on. So it is relatively simple circuit, but still, if you think about how this circuit compute the sort of inputs from the photoreceptors, it's kind of actually complicated. For example, if you imagine red cones are activated, then that's going to give the input to H1 horizontal cell. Then this horizontal cell send the feedback, inhibitory feedback to 3 photoreceptors. So it's not just inhibiting red, but also inhibiting green and blue, which then now inhibited was connected to H2 horizontal cell, which is going to then activate it and then send the inhibition to a different kind of inhibition to blue and UV. So like if you think about the network, it actually is quite complicated. So then what we wanted to understand is how the inputs to cone photoreceptors, which is basically the absorption spectrum opsins, are transformed in the outer retinal circuit to give rise to output of cone photoreceptors. And to do that, we first measure the output of cone photoreceptors using the technique using the 2-photon microscope combined with the calcium indicator, especially in this case, the synaptically targeted GCAMP6F. So this is an example of the PSI-GCAMP6F expression specifically in the UV cones. And then because this is a synaptically targeted GCAMP, it only localizes at the axon terminal of cone photoreceptors. But and then to look at the sort of a spectral tuning of cone photoreceptors, Philip Barter, made a very nice visual stimulator that consists of 14 LEDs with different wavelengths. So there is like that covers the wavelengths from about 350 all the way to almost like far range. So then we can stimulate the opsins, all the opsins that exist in the zebrafish. And then we didn't go shorter than 350 because the shorter than 350 wavelengths is going to be observed by the optics of zebrafish, like such as lens. So then they are not going to see the shorter than 350 wavelengths anyway. So using this visual stimulator, we basically recorded the calcium response of each cone photoreceptor type. So this is an example of calcium imaging. Let's see if I do this. There's a movie. Yeah, okay. So then this is a color of flash. And then you can see that when the lights hit, calcium goes down because the cone photoreceptors are off cells. And then this is a sort of an example trace of the red cone response with different wavelengths of light. And as you can sort of see, and as we expected, the red cone responds strongly to the sort of greenish color because actually the opsin spectrum absorption peak is shifted towards green for zebrafish. However, we did the same recording in green, blue, and UV cones, and then here we start to see something interesting. So, okay, now I was trying to create a movie, but anyway, so the movie is not working. But anyway, so I think it's easier to see it in this calcium trace. So what we found is that for green cones, for example, they actually respond to a different polarity than the off. So like what you would expect is this off response, right? Whereas for green cones, they actually have their own response to a longer wavelengths, whereas there's an off response in the shorter wavelengths. So this is sort of like probably coming from the inhibition. Right. And then blue cone also have this own response in sort of mid wavelengths, whereas the off response in the short wavelength. And then the UV cones are always off response. So there are kind of a variety of response characteristics depending on the cone photoreceptor types. And then we did this kind of recording for many cones from across the retina. So this is each line indicate the single cone, and then this D and AZV indicate the different region of the retina. So this is these, the dorsal and nasal, and the AZ is actually the acute zone. So this is something I'm going to talk about a little bit later, and then ventral. So 4 different regions of the retina. Although we did not see much difference across the region, so we sort of like average all response tuning curve of all cone photoreceptors. And then this is the result. So as I kind of already showing as an example, from the example, the red cone responds strongly to the mid wavelengths. then not much to the short wavelengths, although there is a little bit response to the UV light. Whereas green cone has opponency, it's called, so the on and off, which is called opponency, and then blue the same, and then UV does not have the opponency. So then first thing we ask is, so how does this response tuning curves are shaped by the retinal circuit? And the obvious candidate would be the horizontal set. So now we know like this is the inputs to the photoreceptor, and then the circuit is gonna, outer retinal circuit modify the input to give rise the output from the cone photoreceptors, right? And then if we block the horizontal cells pharmacologically, then what we saw is that now the cones do not have opponency. So this solid line is a response tuning curve of cones after horizontal cell block, whereas the dashed line is with a horizontal cell block. So as you can see, like the cones lose all the opponent components. Whereas for example, red cone or UV cones, the response tuning curves are almost unchanged. So that means that the horizontal is the one that shapes the tuning curve. And then if I overlay the opsin spectrum curve on top of the cone response tuning curve, it matched quite well. So which means that the horizontal cell network is the major component that shapes the tuning curve of the cones. We also tested if there's any components from gap junction connection. So what we did here is that we ablated the UV cones specifically using a genetic method, and as well as we also blocked the horizontal cell. So now what we see is that only there should be no response from UV cones, right? So then after UV cone ablation, what we saw is this solid line. Basically, we still see some response from UV light range. So which means that this UV small response to UV light is not coming from UV cones through gap junction. So this is actually the response characteristics of red cones. Right, and then similarly to blue cones, they also respond to UV light strongly, and then if that remains the same after UV cone ablation. So we think that there's not much contribution from gap junction connection, at least from UV cones. Anyway, so then we further actually. So we now know that this input is shaped by the horizontal cell network, not the gap junction, to give rise to output. And then what we also show is that, so I'm not going to talk about, but what we also show is that we use the computational modeling to demonstrate that the horizontal cell network, especially having these 3 horizontal cells, can actually give rise to this output shape by tuning the connectivity of the horizontal cells. So then next question we ask is why then cones needs to have the spectral tuning curve in this shape? So then to look at, to understand it, we look at the visual scene of the zebrafish. So then Nora Nevara, who used to be a PhD student in Tomsa, flew to India and took an image in the Ganges River. And then now what she did is not just taking the image using the typical GoPro camera. She used the spectrometer to measure the spectral composition of the natural scene. And this is kind of example from one scene. And what we can do is that how the scene is look like by different opsins. And this is using the red, green, blue, or UV opsins and how the scene look like. And as you can see, actually all the visual scenes looks very similar because the opsins are the opsin spectrum overlap quite extensively. So then actually it's not that useful to have 4 different kind of options. Whereas if we can actually mathematically compute like what is the best way to decode the spectrum tuning curve of options, and then this is the result. So using the principal component analysis, we can compute what's the best way, right? So then the PC1 is the achromatic channel that basically respond to the luminance, whereas the PC2 has this opponency, which is the color, and the PC3 is the secondary color channel. And then using these 3, you can see that this particular thing looks very different in different channels. Right, so then we compare this physical component with the major tuning curve. And then what we kind of immediately notice is that the cone spectral tuning curve is quite well matched with the principal component, especially like a principal component 1, quite well over matched with the red cone tuning curve, and then so on for PC2 for green. And PC3 does not really over match with any of the tuning curve, although kind of similar to blue, but slightly different. But we actually further show that this PC3 can be recaptured by combining the blue plus green by flipping green. Anyway, so that's kind of detail. But then just to summarize the first bit of the talk, we can found that what zebrafish photoreceptor do is the splitting the color information into 3 principal components. So red is the PC1, green the PC2, and the blue probably combines the green cone in the downstream circuit to create the PC3, because we know that this PC3 exists in some in the brain, the zebrafish. So there is some downstream circuit have this computation dealing with it. And I haven't talked about this UV channel, but we think that this is the secondary achromatic channel. I'm going to briefly talk about in a minute. And then now, if you look at the mammals, they only, so the most of the mammals have 2 types of cone photoreceptors. They lost the blue and the green during the evolution of the mammals, and then most of the mammals have 2 cone photoreceptors, such as mouse and rats. They are called L and S cones, but genetically S cones are sort of ortholog of UV cones in zebrafish, and the L cone is also a red cone in zebrafish. And then we think that the mammals retain the 2 achromatic channels, because that's the one that's the most important, and those the color vision. However, during the primate evolution, they duplicate the red cone to create green cones. So evolutionary green cones in humans are actually also of red cones in zebrafish. And then they recaptured the color channel, as I talked about in the introduction, to have the primary and the secondary color. So then to do that, now there are lots of study about the human color vision circuit, and we know like there's quite complicated circuitry to create the primary color and the secondary color. But I think that from here, what we can see is that what the retinal circuit is trying to do is to create the PC 1, 2, 3, which has this achromatic and then 1 biphasic primary color and then triphasic secondary color. And then maybe depending on the species, the circuit is wired in a slightly different way because the composition of the photoreceptors are different. But I think the basic purpose of the color vision is still conserved across species. Now I'm gonna sort of briefly talk about the second part of the feature of the vision, which is about the high acuity vision. So, and then this is actually coming from by trying to understand the UV vision, and then now kind of we ended up in the surprising finding, which is that zebrafish do have the high acuity vision area in the retina. So first, we wanted to understand what UV vision do for larva zebrafish. So then what we did is to record the movie of the fish tank using the two filters. One is the UV filter, and then the other one is yellow filter. So then in yellow filter, what you can see here is that what you will see by ourselves in the water, if you dip yourself into the sort of like, the pond or something, you can see rocks, you see the surface of the water, you see the reflection of the rocks on the surface and so on. Whereas if you look at the UV channel, what we notice immediately is that we don't see much, which is kind of surprising. And then this is actually makes sense because the water reflects UV light a lot. So then, and UV light is scattered in the water so that we don't see background much. What we instead can see is that the gradient of light, so the bright UV light from the surface, and it's darker in the bottom. But one thing we notice is this. Again, I'm trying to record it. Play the movie. Let's see. Oh, no. Sorry. Okay. Now it doesn't. Anyway, so then what I wanted to show is that in the UV channel, what we could see is that maybe you can notice here, there's small dots here. And this is actually the zooplankton in water, which is the actually prey for larva zebrafish. And then what's nice about UV channel is that because they don't have much background structure, it's easier to spot small dots. And especially if it's moving, you can easily spot. Whereas in yellow channel, there's so much background details you can see, then spotting the small dots are much harder. So we thought that UV vision is used for prey capture. And first thing we also look at is, next thing we look at is the distribution of UV cones. And what we notice is that actually the density of UV cones are not uniform across the eye. So there is a particular region in the retina, temporal ventral region, that has the higher density of UV cones. And this region actually is looking slight front of the zebrafish, slightly upper from the horizon. And this is how it looks like in the visual field, projecting the visual field. So if there's a fish, then they have this UV cones high density area is projecting the center of the animal. And then we also noticed that the shape of the photoreceptor is different. So this temporal ventral region of the photoreceptor has longer outer segments, not just the density, but the outer segments longer compared to the rest of the eye. So this is the quantification across the eye. So this is the area we call now the acute zone. And then we also tested if the UV cones are important for the prey capture using a behavior assay. So then what we did is actually Neuro did is that mounted the larva zebrafish and agar and they put paramecium. And if fish see the paramecium, they exhibit a stereotypic response, which is eye convergence and then flick the tail. So then we counted this response under UV light or yellow light. So this is indicating this color. And then we alternated the UV and the yellow color every 1 minute. So then, as you can see clearly, the larva zebrafish respond to paramecium under UV light a lot, and then, but almost nothing under yellow light condition. And then, just to confirm that this is made by UV cones, we genetically ablated UV cones, and then this response disappeared. So that means that UV cones are used for paramecium, detecting paramecium, and this is done actually Yeah, so then this is actually quite impressive task to do that because the larval zebrafish eye is quite small. So if you look at the size of the receptor field of single UV cones and how it's spaced out, it is about like 2 degrees apart. Whereas the paramecium would appear as a 1.8 small dot. So which means that when larval zebrafish detect the paramecium, they detect using the single UV cones, which is quite impressive task to do that. And then I'm gonna kind of skip over this part because I'm running out of time, but we went on to confirm that the UV cone can respond to such a brief light flash of UV light using 2-photon calcium imaging. Sorry. And then also we look at the response characteristic of UV cones in the different region. And then what we noticed that in the acute zone UV cones have the slower recovery rate after the response to the brief flash of light, which kind of helps for the downstream circuit to have the higher signal to noise ratio because now UV cone acute zone can can integrate signals more. So then what we did is how this could happen. And then what we found is that the calcium level is higher in the acute zone UV cones, which is quantified here. So this is the CAMP6 signal in the live animals. And then by just looking at this picture, you can see like the signal is much higher here. And this is not due to the different expression level of the sensor, because when we fix this animal and then immunostain for the CAMP using the anti-GFP antibody, signal intensity is more or less uniform. And if we divide this signal by the signal from the immunostaining, the signal is higher in acute zone compared to the rest of the eye. So we think that the calcium level is higher in the acute zone. And then we also look at the molecules that might be responsible for this higher calcium level. And then we found that the many molecules involved in the transduction cascades have the differential expression level in acute zone compared to the rest of the eye. And then we use that expression level difference, and then using the phototransduction cascade model, we can actually recapture how this higher baseline arise by having the differential expression level of the phototransduction cascade. So I'm sorry for this kind of like going through this part very quickly, but this is just a summary. So what we found is for the UV vision in zebrafish is that they have a special region called the acute zone in the ventral temporal region, which is used for prey capture. And then not just the density is different, but then the length of the outer segment is also different. So the acute zone has longer outer segments. And then also the physiology is distinct. And those features are also actually found in human or primate fovea. So fovea is located in the center of the eye, which is actually looking forward. Same for the larval zebrafish. And then the density of cone photoreceptor is higher in the fovea. And then similarly to zebrafish, they have the longer outer segments as well as slower response kinetics. In this case, the time to peak is slower, whereas for zebrafish, what we found is the recovery is slower. But essentially, we think that it serves for the same purpose, so integrate more signal. And the even more interesting thing is that the phototransduction cascade molecule, GNGT1, is specifically expressed in the fovea cones, which is one of the molecules we found as a differential expression in zebrafish acute zone. So what we kind of saw is that there are many aspects that are similar between zebrafish high acuity area and the human fovea. So now I want to move on to the last bit, which is the unpublished part. So then hoping to spend a little bit more time. So the night vision circuit, which is called the Rod pathway, is quite well understood in the mammals. So this is just a diagram, but this has so this part of the study is actually has started when I was in Rachel's phone lab, and it took for a while, and we had many collaborators alongside while doing this project, and then we are hoping to be able to publish pretty soon. So this is about the Rod pathway. And the Rod pathway is kind of fast described by Ramon Cajal. He noticed that there are two types of photoreceptors. One is rods, one is cones. And then he described that there is a specific bipolar cells that only connect with rods. And this is over 100 years ago. But then as the more technology developed, then the Hope and the other people describe the cell types that are involved in Rod pathway, which is about 50 years ago. There's a Rod and Rod bipolar cells, and then there are two types of micron cells called A2 and the 17th. They form the stereotypic circuit, and now we know that this diagram is quite conserved across mammals, so the rods connect to Rod bipolar cells. And then these Rod bipolar cells do not rarely connect to the output neuron ganglion cells, but rather connect to these two types of interneurons called A2 and A17. And the A2 amacan cells now connect to cone bipolar cells, which then give output to ganglion cell. So the major component of this Rod pathway is the Rod bipolar cells. That's where the Rod pathway starts. And then the recent paper by Josh Han in Kaushik's lab basically identified found that the bipolar cell types in mammals are more or less conserved. So this is sort of the figure from his paper now in bioRxiv. So by combining different species across mammals, they could now cluster into distinct types. And then Rod bipolar cells here, which is conserved across all mammals. Right? And also, so that means that the molecularly Rod bipolar cells are different from the rest of the bipolar cell. And then we also know that the connectivity to photoreceptors are different. So rods, so there are about 15 bipolar cell type in mouse case, and then Rod bipolar cells are mostly connected rods. So traditionally, historically, many people thought that the Rod bipolar cell only connect the rods, but we now know that actually they do also connect with a few cones. Right? So, but then the most distinct part is that they connect with pretty much all rods within the reach of the bipolar cell. Whereas other bipolar cell, many of them do not connect with rods, or even some of them do connect with rods, but the number is much fewer compared to the available rods within the reach. Okay, so the point is that Rod bipolar cell is quite different from the rest of the bipolar cells. So that is the what we know in mammals. So what about non-mammals? So this is the connectivity, so anatomical connections in zebrafish across different bipolar cells. So what we know is that many bipolar cells actually integrate input from rod as well as cones. So like there are 3, 2, and then 3 and 1. So total 5, 9 5% at least here. So then what we know about non-mammary bipolar is that the majority of the bipolar actually integrate input from ROT as well as cones. And then also we haven't found, no one found that we don't actually know then about the downstream of the ROT bipolar. Anyway, so then what we ask is that, is there any bipolar cell that are specific for ROT pathway in zebrafish? So first thing we did is to look at the molecular classify the bipolar cell using the single cell RNA sequencing. So this is done by Yvonne in Hervik Bayer's lab, and then she did it the single cell RNA sequencing experiment in Joe Sain's lab. So using this transient line, which express the GFP in all bipolar cells. So she dissected and dissociated and factorizated the bipolar cells, and then did the single cell RNA sequencing. And then Josh Hans did the analysis part, and then he identified total 23 cluster of bipolar cells. And from this, we can sort of like classify bipolar cells into different groups. For example, on and off bipolar cells using them by looking at the expression of the marker genes known for mammals. So for example, IL-1 is a marker for on bipolar cell in mammals, and then GNB3 is the same for that. And then we could kind of see some of the bipolar cells highly express on markers, and some express off markers, which is a Greek one in this case. So we could sort of like classify into different groups. And what we kind of found here is that, and then we surprised about is that we saw 2 bipolar cells that express both bipolar cell marker. So which is called the GRAM-D1B. And then these bipolar cell, one of them strong PRKC, which is another marker for bipolar cell, not the other one. But then overall, these 2 bipolar cells are sort of similar to each other. And this is also shown here. So it's called the dendrogram. So what it's shown here is that the similarity of the gene expression patterns across bipolar cells. So if you see 2 bipolar cells across each other, means that the genes expressed in these 2 bipolar cells are similar to each other compared to the bipolar cells farther away. Right? So then what we can see from, for example, mouse, is that the bipolar cells are nicely separated into 3 groups, 1, 2, 3 here. And then these 3 groups correspond to off bipolar cell, on bipolar cell, and both bipolar cell. So when we look at the zebrafish, we immediately notice that these 2 bipolar cells are separate from the rest of the bipolar cells. So it kind of also made us think that maybe zebrafish has 2 ROT bipolar cells here. Right? And then the rest of cones, what we kind of saw is that the on and off bipolar cells are not well separated, which is so then it made it difficult to sort of make a guess which one is which one. But the fact that these 2 bipolar cells are separated from the rest, and then these 2 express the ROT bipolar cell markers, we hypothesized that these 2 are ROT bipolar cells, although there are 2 of them compared to the only one in mammals. And this is also supported by the findings we made in parallel, which is that we saw this transient line, which expressed this cellular fluorescent protein in specific bipolar cells. And this bipolar cell has the PKC expression. And then if you look at the axon terminal, this distributed more or less uniform. And then when we look at the dendrite, they form like a nice sort of tiling, although there are some like a blank spot because the expression of this transgenic line is not uniform. So which indicate that this tiling indicate that this biparcel is a single type of biparcel. And then this axon stratified at the bottom dipole, which is another morphological feature of the biparcel. So we saw like, okay, so now probably this transgenic line is labeling a rod biparcel. What we also found is that there is another transgenic line which labeled the biparcel with the axon terminal at the bottom dipole. But then these biparcel are not PKC positive. And then if you look at the axon terminals, they are more or less uniform. And then also dendrite formed tiling. So which indicate this biparcel is also a single type of biparcel, but different from this biparcel label in VSX1 because they are not PKC positive. So now we found the two bipolar cells that has a morphology very similar based on the axon terminal stratification. So we suspected that these two bipolar cells correspond to the two rod bipolar cells we identify using a single cell RNA sequencing. So then to confirm that, we look at the expression of the marker genes. So from the single cell RNA-seq, we can identify genes that are specifically expressed in one of the rod biparcel, but not the other one. So using this marker, so one is the S10810B, and then the other one is UTS1. We found that the S10810B is specifically expressing VSX1 positive biparcels, as you can see by the overlay of this, whereas the UTS1, which is a marker for 19, is or expressed in the VSX2 positive biparcel. So then we next look at whether these two biparcels are actually indeed connected with rod using the immunostaining against rod as well as different types of cones. So now this is the side view of the biparcel, and then rods are labeled in cyan, and then red cones are labeled in red here. And if you look at the dendrite, so we can actually see which photoreceptors this biparcel connecting by looking at the invagination dendrite, dendritic tip in the photoreceptor invagination. So then these biparcel dendrites are connecting these rod terminals as well as red cones. And this is actually the same for this VSX2 positive biparcel, as you can see here, and this is quantified in this plot. So each on average, each biparcel connect about 40 to 50 rods, and then fewer red cones, about 10 red cones. So this means that these two biparcels are indeed receiving lots of input from rods, right? And a little bit from red cones. So this is similar to mammalian rod biparcels. Although this majority of the red, the reason they have the dominant input from rod is not because they are selective for rod over red cones. Actually, they do grab all rods or red cones within the reach. So if we count the percentage connected within the dendritic field per biparcel, it's almost 100%. So because the number of rods are still numerically higher than red cones, then these two biparcel are dominant input from rods. So now, just to summarize what I talked about up to this point, we found that there are two rod biparcels, unlike mammals, But then what about the downstream circuit? To look at this, we use the serial electron microscopy reconstruction of the rod bypass pathway. So here is an example slice of the zebrafish retina, and then we can identify the stereotypic big bypass axon terminal in the bottom of the IPL. And then by reconstructing the axon terminal, we can see two types of axons. One has the ribbon, which is the synaptic location of the bypasser, only exists in the terminal, or the other bypasser has the ribbon in the terminal as well as somewhere in the shaft. These actually correspond to RBC1 and 2 because by using the rebar immunostaining for the ribbon synapse in the bypasser, we see that RBC1 only has rebar in the axon terminal, whereas RBC2 has a ribbon in the shaft as well as the terminal. So we can distinguish the RBC1 and RBC2 based on the localization of the rebar. And we also confirm that these axons with the big axon bypasser with the big axon terminals indeed connect with rods by tracing all the way up to dendrite, and then we confirm these two bypassers connect with lots of rods. Okay, so now we reconstructed the all those bypassers with large axon terminals and assign the RBC1 and RBC2 based on the ribbon distribution in the volume. And then, as you can see, these two bypassers, RBC1 and 2, are more or less uniform. So we think that we managed to label probably all the RBC1 and 2 in the volume. So now we then focus on two of the bypasser, one RBC1 and one RBC2 at the center of the volume, and then trace all the postsynaptic neurons here. Right? So then there's a bunch of postsynaptic processes here. Okay, so then we sort of like gonna tease out. So what kind of postsynaptic neurons these RBC1, RBC2 are connecting with? So let's focus on the RBC1. What we notice is that the most of the postsynaptic neurons are amacrine cells, and then very few with ganglion cells. This is kind of similar to the mammalian rod bypass cells. And then the number of postsynaptic sites, synaptic sites are varied depending on the neurons. So most of the neurons receive 1 or 2 or maybe up to 4 synaptic input from one RBC1, except of this one amacrine cells, which received 14 synaptic inputs, which actually then occupy about 1/3 of all synaptic sites of this particular RBC1. Most of the amacrine cells are monostratified, and there are only 2 multi-stratified amacrine cells we found, although one of them is the one that received lots of input from this. Among these monostratified ones, we found 2 types based on the stratification depths. So one type stratifies slightly upper than the rest of the group. And not just the stratification level, actually, we noticed that the synaptic arrangement is different. So one type of amacrine cell only received input, but never gave the feedback input to the same rod bypasser. Whereas this one always formed the reciprocal synapse, which is called, they receive the input, they also give the feedback back, which is called the reciprocal synapse. And this is an example here. So this type stratifies at the bottom of the IPL, give the reciprocal synapse. And then the other type of amacrine cells stratify slightly above, and then there's no reciprocal synapse and only receive the input. So from here, basically, the most of the synaptic output goes to 3 types of amacrine cell. One is the non-reciprocal, one is with reciprocal synapse, and one is a multi-stratified amacrine cell, although we only found one this amacrine cell for this particular RBC1. So what about the RBC2? Again, we did a similar analysis. Most of them are amacrine cells, although the number of sites are fewer. So like most of them receive, 1, 2, up to 4, and we did not find any amacrine cell that received lots of input. Most of them monostratified, and then they are all stratified in the same level, and then they always give the reciprocal synapse, except this one amacrine cell. So for RBC2, we think there's the most of the synapse goes to one type of amacrine cell, which is a monostratified reciprocal type. Okay, so now we know about the postsynaptic neurons for mammalian pathways, right? So which is called A17 and 2. And then when we look at this postsynaptic amacrine cell type for RBC1, we sort of notice the similarity between two. So one type gives the reciprocal synapse, which is sort of similar to A17. And then the other feature actually about A17 is that they only receive synapse from rod bypass in mammals. And we also found the similar specificity in zebrafish amacrine cells. So this amacrine cell only receives input from rod bypass. And then there's another type of amacrine cell in the rod bypass called A2, which we also received lots of input from single rod bypass cell, often more than 20 snaps from one rod bypass cell. And this is similar in zebrafish multi-stratified amacrine cells. But there are other features. So, for example, A2 amacrine cell receive input from rod bypass cell, but then give output snaps to cone bypass cell, right? So we ask if this feature is also conserved in zebrafish. So we, as I mentioned, we only found one multi-stratified amacrine cells. However, when we look at the another rod bypass RBC1 in the neighboring one, then we could easily find amacrine cell that receive lots of input from the neighboring RBC1, and we trace this amacrine cell, and then we indeed found two multi-stratified amacrine cells next to each other. So here is a side view of these two multi-stratified amacrine cells, and we map the synaptic location for these two amacrine cells. So the input from bypasses indicated in magenta here, mostly localized in the bottom of the IPL, whereas the output, the bypasses, are only exist in this upper layer of the IPL. And then we also noticed that there's this process, lots of processes that contact with bypasses, but they don't form synapse. So, and this is very similar to what we know about the mammalian A2 amacrine cells. So they receive bypass input in the bottom of the IPL. And then they also form the gap junction connection with on-cone bypass cell, which is indicated here. So for our case, we could not resolve the gap junction because of the resolution limit, but we suspect that this non-synaptic contacts probably gap junction also needs to be confirmed. And then this amacrine cell also give output only in the upper layer of RPL. So we see like lots of similarity between these two. And so this is a summary for rod pathway. So what we found is that we found two rod bypasses, and the one type has the circuitry, very similar to mammals. So then just to summarize, so as we studied the Western circuit for zebrafish, we found lots of similarity between human and zebrafish, including color vision, high vision, and night vision. So then this is quite interesting because if you look at the evolution tree of the vertebrate, mammals and the fish are quite far away, and there are even amphibian reptile bars in between. But the fact that we found many similarity between mammals and fish means that those, at least we know that these bypass cell, vertebrate retinas probably share the similar features. And I want to end my talk by just emphasizing the fact that we know a lot about mammals, especially this is the current trend of the modern animals in the retinal research, and many of the research use the primate human because those are mostly like a clinical research. And for the basic research, the mouse is the major model animals. So this is the number of the publication across the different year. And then if you look at the number of paper using the zebrafish, this is very tiny. So I think that now we know that zebrafish share many similarity with human vision. We can kind of make use of the zebrafish a lot more and understand how the human vision works. With that, thank you for your attention.
Thank you very much, Takeshi, for this very rich in information and also unpublished stuff presentation. So in the interest of time, I'm already going ahead and posting the Zoom room link that we're currently using in case people want to join us. And as a reminder to the audience, after a first brief round of public moderation, we will be continuing offline with people that are interested. And when I terminate the live broadcast, the link will disappear. So make sure you click it if you want to be part of this now. As we are waiting questions in the chat, one question that I have is like, you mentioned 2 types of bipolar cells, right? And do you know anything, or do you expect something about their polarity? Do you expect the same polarity, like both of them to be on cells or off?
Right. So that's a good point. I skipped that data. We did the electrophysiology from these 2 bipolar cells, and we confirmed those 2 are OM type bipolar cells, similar to mammalian bipolar cells.
The first question in the chat is from Tom Baden. So birds have a transcriptomic signature for a rod bipolar cell, but no obvious anatomical counterpart. Also, unlike in fish, they co-wire with double cones rather than red single. What's up with that? Do you have any ideas?
Right. Yeah. So I guess we are still lacking lots of information from those species. So it's kind of hard to tell at this moment. And then also, even for zebrafish, right, the molecular, if you only look at the molecular expression, it is kind of hard to guess which one is the rod bipolar cell, right? We were kind of more, sort of by looking at the connectivity of the downstream and the dendrite and then combining the molecular expression, then we kind of start to guess like, okay, this is probably the rod bipolar cell in zebrafish. So I think, we can make a guess, but at this point, it's really hard to say why there's no similar bipolar cell or not.
Right,
different species.
But the fact that you observe some, like the abundance of synapses is with rods, and then you also have a lot of reds compared to other photoreceptors. Is that as expected or not really?
Sorry, say that again. So
that you have when it comes to cone photoreceptors, you have a lot of red cones and not like blue, green.
Oh,
whatever.
I see. Yeah.
And like their spectral curve is kind of similar between rods and red cones, right? So would you expect that this would be the reason behind it, or it's completely unrelated?
Right. Well, I can only speculate. But the first part of the talk, I talk about what cones are doing, right? And the red cones are achromatic signal channel. And this is probably the reason why this rod bipolar cell connect to red, because rods are also achromatic channel in the dim condition. So my guess is that this rod bipolar cell in zebrafish, by connecting rod and red cone, they have the achromatic channel through different light intensity. But something to be confirmed.
Right. There is another one from Tom appearing in the chat, and I think after that we will have to probably terminate the live broadcast and continue offline with interested people. So I think he's trying to test what you know about different animals. So the question is, why do diurnal lizards retain a rod bipolar cell signature despite not having rods?
Diurnal lizard.
Yeah, so he says that
Okay.
they have a rod bipolar cell signature,
How,
but they
okay.
don't have rods.
Right. Well, I guess again, like, you know, how do you define the rod bypass signature? If you look at, you know, PKC expression, for example, well, that's just one molecule. So my guess is that, you know, maybe these bypass cell types are plastic in some sense that, you know, maybe they can serve. Or no, actually, that's not true, right? So, like, for example, if we think about like a zebrafish rod bypass cell, they connect rod and recon. So if we imagine if they don't have rods, they just connect to recons and then they still serve the same thing, like achromatic channel.
Mhm.
But it's just a guessing, you know, I'm just guessing.
Yeah. Given that Tom is already among the people here, maybe we can continue this offline. I will go ahead and post the Zoom room link one last time so people can make sure to copy it right now, because soon, as the video will, you know, upload on YouTube, the link will not be visible. So thank you very much once again, Takeshi, for this lovely talk. And I can't wait for the questions that will happen now. Personally, I have way too many.
Yeah.
See you.
See you.